Hello everyone, I'm Snelly Sofia, a second year doctoral student in the Biology and Health Laboratory of Ibn Tufayl University, Faculty of Sciences, under the guidance of Professor Akhwairi Omar. I'm delighted to introduce to you this video presentation titled Exploring the Correlation Between Gut Microbiota and Hypertension Towards New Perspectives in Cardiovascular Health. In this presentation, I will talk about the context of our research, previous research in order to define our objectives, material and methods that we will explore, results that we want to achieve, and at the end, I will give a small conclusion. Hypertension, also known as high blood pressure, is a silent but potentially life-threatening condition that affects millions of people around the world. Let's take a moment to explore the gravity of this health issue. It is estimated to cause 9.4 million deaths worldwide each year. The World Health Organization estimates that in 2025, there would be 1.5 billion people with hypertension and 7.4 million deaths due to hypertension, or about 12.8 of the annual total. In fact, its leading cause the cardiovascular diseases, including heart attacks and strokes. In Morocco, the situation is not different. Hypertension is growing concern. The Moroccan Ministry of Health reports that 89,786 people with hypertension were diagnosed in 2021. 61% of them are women, and it's estimated that many more remain undiagnosed. Now that we understand the seriousness of hypertension, let's explore the current approaches for managing this condition and the challenges they bring. Traditionally, when hypertension is diagnosed and depending of its severity, doctors direct their patients towards salt-free diets or prescribe medications. These medications, including diuretics and beta blockers, can be quite effective, but they also with potential side effects, which can cause kidney problems. Additionally, this treatment often requires a lifelong commitment, and not all patients react the same way to medications. It will take time to find the right combination for each patient. Over the last few years, a new perspective has opened in our understanding of what goes in our organisms. Bringing the light an unexpected actor, the bacteria that populates our intestines, known as intestinal microbiota. Thanks to scientific progress, this microorganism known as the rule of digestion and immunity have been defined as the main factor favoring the onset and development of several diseases such as diabetes, obesity, autism, and even hypertension. Previous studies carried out in other countries, such as China and Finland, have proven the existence of a correlation of the intestinal microbiota and the development of hypertension. An analysis of the 16S ribosomal RNA gene showed that the human intestinal microbiome in patients suffering from hypertension harbored a lower microbial diversity than that of the intestine of a healthy individual. Bacterial germs have been correlated with the development of hypertension, such as Enterobacteria and Escherichia shigella in the Chinese population and Streptococcus in the American population. The intestinal microbiota has therefore been shown to be an essential factor in the regulation of blood pressure. Despite the growing importance of the gut microbiota in global health discussions, no study to date have specifically examined its role in hypertension within our nation. Our project aims to fill this gap, paving the way for fresh insights and novel perspectives on cardiovascular health. Our aim is to analyze the composition and diversity on intestinal microbiota in hypertensive individuals in Morocco compared with healthy subjects, determine whether there is a correlation between the existence of certain bacterial species and the development of hypertension, established link between the composition of the intestinal microbiota and the inset and development of arterial hypertension and the risk factors associated with this disease. To achieve our objectives, we conduct a prospective longitudinal study over 3 years by recruiting a group of 200 patients with hypertension and 100 healthy volunteers. We will collect stool samples from the participants and then analyze the gut microbiota by sequencing the V3, V4 hypervariable region of the bacterial 16S ribosomal RNA gene. The bioinformatics analysis of the results obtained will allow us to identify the bacterial species present in their intestines, followed by statistical analysis to determine existing correlations. We anticipate that the statistical analysis of the results obtained will provide evidence of the existence of significant differences in the composition of the intestinal microbiota between hypertensive individuals and control subjects, identify potential association between specific microorganisms and the severity of hypertension among the Moroccan population. In conclusion, our research is a crucial step in understanding hypertension and its link with the gut microbiota. We are convinced that the results of this study will have a significant impact on public health and preventive medicine in Morocco. Thank you for your attention.